Dear viewers, greetings. In this video, we are going to see the major difference between exotoxin and endotoxin. First, exotoxin is defined as the proteins produced inside the pathogenic bacteria as a part of their growth and metabolism. And the endotoxins is defined as lipid portions of lipopolysaccharides that are the part of outer membrane of bacteria. Mainly gram-positive and gram-negative bacteria act as the source for the exotoxins, but gram-negative bacteria alone act as a source for endotoxins. Exotoxins are secreted by living cells, but endotoxins are the integral part of cell wall and secreted by lysed cells. Regarding the secretion, exotoxins are secreted out of the living cell, but endotoxins not released outside the cell until death of the cell. Chemically, exotoxins are made up of protein or short peptide, and endotoxins are made up of lipopolysaccharides protein complex. The molecular weight of the exotoxin is 10 kilodaltons and the molecular weight of the endotoxin is 50 to 1000 kilodaltons. The chromosomal location of the exotoxin is extrachromosomal genes or plasmids, and the location of the chromosomal location of the endotoxin is chromosomal genes. And next, components. The exotoxins are composed of two subunits, that is subunit A and subunit B. The A subunit is seen to have catalytic activity, whereas the B subunit is required for binding with an appropriate cell receptor. Endotoxins are composed of three basic components. The first basic component is O antigen. The second basic component is core oligosaccharides, and the third basic component is lipid A. The toxicity of the exotoxin is high, but the toxicity of the endotoxin is low. Cell lysis is not required for the extraction of exotoxin, but the endotoxin's extraction requires the cell lysis. The exotoxins are heat labile. They will be degraded at 60 to 80 degrees Celsius, but endotoxins are heat stable for 1 hour. It will be able to withstand the temperature up to 250 degrees Celsius. And next, boiling. Exotoxins are denatured on boiling, but endotoxins are not denatured on boiling. Regarding the toxicity dosage, exotoxins are very active in minute dosage. They are highly toxic, fatal in microgram quantities. But endotoxins are active only in large dosage. Endotoxins are moderately toxic, fatal in microgram quantities. Next, the mode of action. The mode of action of the exotoxin includes enzyme-like mechanism, and the mode of action of endotoxin includes tumor necrosis factor and interleukin 1. And fever producing ability. No fever production was observed in exotoxin, but endotoxins produce fevers by induction of interleukin 1 production. And the effect of exotoxins on the host is variable in cells or tissues. That is mainly depending on the host, maybe cytotoxin, neurotoxin and enterotoxin. The endotoxin produces fever, diarrhea, vomiting, lethargy, malaise, shock and blood coagulation in the host. Next, the antigenicity. The antigenicity of the exotoxin is strong, but the antigenicity of the endotoxin is weak. Regarding the neutralization by antibodies, exotoxins are neutralized by antibodies, but the endotoxins are not neutralized by antibodies. And the enzyme activity, the exotoxins mostly has enzyme activity, but in endotoxin, enzymatic activities are limited or absent. Enzymes like hyaluronidase, collagenase, certain protease, nuclease, neuraminidase and phospholipase A are present in the exotoxin, In endotoxin, enzymes like catalase, fibrinolysin, IgA protease and IgG protease are present. Exotoxins are very specific in nature, but endotoxins are not very specific in nature. Exotoxins usually binds to the specific receptors, but in endotoxins, specific receptors are not found. Exotoxins are specific to certain bacterial strains, but endotoxins are not specific to any bacterial strains. Separation of exotoxin from the cultures was done by filtration, and the separation of endotoxin from the cultures was done by cell lysis. Some representative diseases caused by the exotoxins are botulism, tetanus, gas gangrene, diphtheria, cholera, plague and staphylococcal food poisoning. And some representative diseases of endotoxin are typhoid fever, tularemia, endotoxic shock, urinary tract infection and meningococcal meningitis. And finally, a toxoid for immunization will be prepared from the exotoxin by treatment with heat or formaldehyde. But for endotoxin, the toxoid for immunization is not feasible. Dear viewers, thank you for your support. Thank you.